Hello, I am Laskar and I am going to talk about our study on risk factors for melanoma by anatomical site and evaluation of etiological heterogeneity. This study was conducted at the International Agency for Research on Cancer and was led by IARC scientists Dr. Ida Ferreira-Iglesias, Dr. Hilary Robbins, and Dr. Professor Ann Cast at the Faculty of Medicine and Health, University of Sydney, Australia, along with collaborators from the Australian Melanoma Family Study, the UK population-based case control study, and the Genome Consortium. Two main etiological pathways have been proposed for melanoma, which is determined by nevi and the type of sun exposure and are often related to anatomical locations of melanoma. But such studies on risk by anatomical sites are often limited by small sample size and lack of detailed risk factors data. So, in the current study, we examined associations with melanoma by anatomical site for a comprehensive set of risk factors that includes pigmentary and nevus phenotypes, UV exposure, and genetic risk factors. In order to do so, we pulled data from 3,592 participants who were recruited through two population-based case control studies in Australia and the UK and estimated the risk of developing melanomas in different anatomical sites such as head and neck, trunk, upper and lower limbs. Our results show that although phenotypic characters such as lighter hair, eye and skin colors were all associated with increased risk of melanoma, but very fair skin was weakly associated with melanoma on the trunk as compared to melanoma on other sites. Among the most important findings was the presence of many nevi as much stronger risk factor for melanoma on the trunk, upper and lower limbs as compared to melanomas on the head and neck. Also, an increased weekday sun exposure was associated with melanoma on the head and neck, but not on other sites. These findings are interesting and consistent with the dual pathway hypothesis, which proposes that risk of melanomas in the trunk is determined by the propensity to form nevi and intermittent sun exposure, whereas melanomas in the head and neck is more likely to be caused by chronic and continuous sun exposures. Although we didn't find any heterogeneity by polygenic risk scores of melanoma. To conclude, by studying a series of more than 3,500 participants, we found an increased number of nevi and very fair skin were the risk factors that show heterogeneity by anatomical sites of melanoma. To know more about our study in details, please read and share our paper from the British Journal of Dermatology websites in the links below. And thanks for watching.